Welcome viewers. Welcome to the second lecture on plasmids. In today's lecture, we will be covering the points regarding the copy number control of a plasmid, plasmid incompatibility, partitioning of a plasmid, and plasmid addition systems. To start with, let's have a look on the definition of copy number of a plasmid. Copy number is defined as the number of copies of a plasmid that reside inside a bacterial cell just after the cell division. Depending on the copy number of a plasmid, they can be classified into two broad categories. One is called high copy number plasmids. High copy number plasmids means there exists many copies of a plasmid inside the bacterial cell. Example of such plasmids is called Col1 type of plasmids. There are certain plasmids which remain in very low copy numbers, or we may say that only one copy of the plasmid exists inside the bacterial cell. Such plasmids are called stringent plasmids, or example of such a plasmid is F plasmid.
Ma'am, why it is necessary to regulate the copy number of plasmid?
Yeah, this is very much important to regulate the copy number of a plasmid, as because if there is no proper regulation and the replication of the plasmid goes on indefinitely, then the cell will be enormously burdened with a plasmid. On the other hand, if the rate of replication of the plasmid is not at par with the rate of replication of the host cell, then there might arise a situation wherein certain daughter cells won't receive any plasmid at all. That's why this copy number control mechanism is mostly studied by the scientists, and we have gathered enormous amount of information regarding the control mechanisms of copy number of different plasmids. So broadly, we can say that there are three different categories to control the copy number of a plasmid. Number one category comprises of the control mechanism where the control is executed by a counter-transcribed RNA or an antisense RNA, which is responsible for inhibiting the initiation of replication of a plasmid. As we all know that to initiate replication of a plasmid, we need a RNA primer. So, if an antisense RNA is synthesized and it restricts the binding of this RNA primer to the origin of replication, then the replication initiation pathway is restricted. So, this is one way of restricting the copy number. The second way is not only using a counter-transcribed RNA, there exists certain plasmid coded proteins that actually help in controlling the replication rate of the plasmid. And the third process by which copy number can be controlled is involvement of proteins. These proteins are mostly plasmid coded proteins, and they have got binding affinity towards certain repeat sequences called heteron sequences. So on the basis of these ways of restricting copy number of a plasmid, we will take up examples of different plasmids and their mode of copy number control. So to start up with Col1. Col1 is a very high copy number plasmid. Therein we will see that a short stretch of RNA called RNA2 is responsible for binding onto the ORI-V region, that is the start site of the replication of the plasmid. Once this RNA2 binds to the DNA and forms a RNA and DNA hybrid, and then this RNA-DNA hybrid is being acted upon by RNase H. What RNase H does, that it disrupts the secondary structure of the RNA2, and it makes the 3' hydroxyl end available so that the DNA polymerase can come and carry on the polymerization process and carry on the replication process. But there exists a antisense RNA or counter-transcribed RNA called RNA1. What it does, it has got binding affinity towards RNA2, and it forms a complex with RNA1 and RNA2, which is called as the kissing complex, as because the stability of this complex is very less, and it really needs the help of another plasmid coated protein called ROC protein. ROC protein comes and forms a complex with RNA1 and RNA2, stabilizes the complex, and as because this complex is formed, RNA2 is unable to bind to the origin of replication. And if it is unable to bind to the origin of replication, then replication of the plasmid cannot start, and by this way the Col1b plasmids are regulating their copy number. Next, we will talk about another mechanism of regulating copy number in case of R1 plasmids. In case of R1 plasmids, we will see that on one plasmid coded protein called Rep-A is responsible for binding onto the ORI-V region, which is the start site of replication. What it does, it carries on the unwinding of the DNA and it initiates the process of replication, taking help of certain host proteins. In case of R1 plasmid, we have seen that there are two promoters called P-Col1b and P-Rep-A. Both of them are responsible or capable of synthesizing Rep-A protein. But the difference lies that the promoter for Rep-A, which lies in the intervening region of P-Col1b gene, is repressed by binding of Col1b protein. So as soon as the cell is first infected by the plasmid, at first stretch, when replication rate needs to be faster, it gets replicate, or the Rep-A gene gets transcribed from both the promoters, P-Col1b as well as P-Rep-A. But as soon as P-Col1b is synthesized, what it does, it binds to the promoter for P-Rep-A and represses the synthesis of Rep-A protein, as a result of which Rep-A is synthesized in a regulated fashion from only P-Col1b promoter. But once the R1 plasmids reaches their desired copy number, then they really need to switch off the synthesis or expression of Rep-A protein. Under such circumstances, there exists an antisense RNA which is being synthesized from the complementary strand, and that is called as Cop-A RNA. And what it does, that Cop-A RNA hybridizes with the Cop-B mRNA and it restricts the synthesis of Rep-A gene even from the P-Col1b promoter. So this is how we have seen that just by regulating the expression of Rep-A promoter from two different promoters, the copy number of R1 plasmid is controlled at a desired level. Next, we will talk about the copy number control mechanism of Col1b-P9 plasmid. It is a bit different, wherein we will see that the Rep-Z protein, which is plasmid coded and is needed to initiate the replication process, is being translated in the downstream of a leader peptide that is called as Rep-Y. And once this RNA is transcribed, we will see that certain secondary structures like 1, 2 and 3 are formed. And because of the formation of these secondary structures, what we can see that the Shine-Dalgarno sequences for the Rep-Y protein, that is the leader peptide, is exposed and it can undergo translation perfectly. On the other hand, the Shine-Dalgarno sequence for the Rep-Z protein, which is really needed to initiate the process of replication, is occluded. So what happens, we here see that there are certain elements in the loops of structure 1 and 3 which can form base pairs among themselves. So what happens during the translation of Rep-Z protein or Rep-Y protein, what happens that it opens up the structure 2 and it facilitates a pseudo-knot formation between component of structure 1 and structure 3. Because of this pseudo-knot formation, what happens that the Shine-Dalgarno sequence for Rep-Z gene gets exposed. And herein we can see that just translation of the leader peptide is promoting the translation of Rep-Z gene. So this system goes on until and unless the cell or the plasmid reaches its desired copy number. Once it reaches its desired copy number, we will see that another stretch of antisense RNA that is called as synthesized from the ink region, what it does, it forms base pairs or with the structure 1 of the leader peptide. And if the leader peptide forms this stem loop structures are involved in interactions with the antisense ink group, then what happens that their capability to form pseudoknot to facilitate the exposure of Sine-Delgado sequences is lost. And in this way, we can see that the antisense RNA, which is just forming complementary base pairings with the leader peptide rep Y, is restricting the translation of rep Z. So this is the third mechanism which we can see that how the copy number is being controlled. Next, we have got the case of PT181 plasmid, where the copy number control mechanism is a bit different. Herein, what we can see here that there are structures 1, 2, 3 and 4 which are formed, but what happens that as the copy number is reached, the system really wants to switch off the replication of the plasmid. Then it happens that the antisense RNA will bind here and it leads to alteration in the structures which are formed in the mRNA, and this alteration in the structure leads to generation of a premature transcription, row independent transcription termination signal. Herein we will see that there is premature transcription termination or attenuation, which leads to control in the synthesis of a particular gene which is responsible for carrying on the replication. So these are the methods wherein we have seen the involvement of various antisense RNA in regulating the copy number control. And as we were discussing that there are certain mechanisms wherein we will see that certain proteins are involved in restricting the copy number, wherein the proteins are plasmid encoded proteins, and this protein is called, say, rep A protein. What it has got the capability, it has got multiple binding sites in the sequences called iteron sequences. These iteron sequences are stretched within the plasmid, within the ori region, and what it does, it leads to multimerization or formation of dimers between the plasmids. And if dimerization occurs, then this leads to restriction in the replication of the plasmid, and thereby as the copy number of the plasmid is increasing, the number of copies of rep A protein will increase and the probability of formation of dimers will increase. And if dimerization is increasing, then the copy number or the replication rate is decreasing. So next we are coming to another topic called as the curing of a plasmid. Generally cells, there are certain situations wherein we will see that the plasmids are being lost by the cells. This is called as the curing of a plasmid.
Ma'am, is there any mechanism to prevent the curing of the plasmids?
Yeah, there exists many mechanisms to restrict the curing of a plasmid, as because the plasmid doesn't want to get cured, it opts various mechanisms to restrict the curing. So we'll take up those points one by one. The most prevalent mechanism is called as the plasmid addiction system. Second one is called site-specific recombinases that actually resolves the multimers. Actually, multimerization is the driving force towards loss of a plasmid. So plasmid opts for a system to avoid multimerization. And the third one is called as the partitioning systems. They have got certain genes that are involved in partitioning of plasmid uniformly onto the daughter cells. So let's start up with plasmid addiction system. So generally we have seen that there are certain plasmids that code for two different proteins, or plasmid addiction system comprises of two component proteins. One protein is called as the toxin and the other protein is called as the antitoxin or antidote. So what happens, so long the plasmid is residing inside the bacterial cell, the cell produces toxin as well as antitoxin. But the key word is that the stability of the toxin is much more than the stability of the antitoxin. So the moment the bacterial cell tries to get rid of the plasmid, or the plasmids are getting killed, then what happens that the antitoxin gets degraded, but the feeble amount of toxin that still remains inside the host cell is enough for killing of the host cell. So this is the way we may say that the plasmids are creating threat, that if you want to get rid of me, then I am going to kill you. So the next thing of getting overcome the curing of a plasmid is resolution of multimers. As I said that whenever there are certain repeat sequences which leads to the formation of dimers or multimers in a plasmid. So this multimerization is the result of recombination between the monomers, and this subsequent recombination can form higher and higher multimers. And also we have seen that there are certain plasmids which undergo rolling circle mode of replication, and if after during the termination of rolling circle mode of replication, this termination is not done perfectly, then also it leads to the formation of multimers. Then we have seen that if multimers start accumulating inside the one bacterial cell, then during partitioning or division of the plasmid to the daughter cells, it becomes difficult for the system to recognize these multimers as different entities. They consider them as single plasmid, and all the multimers get remained or retained inside one cell, and the daughter cell doesn't receive the plasmid at all. So to avoid this, we have seen the existence of certain sequences within the plasmid, and there are certain genes which are coded by the host cell to prevent multimerization or to prevent or to resolve the multimers. To resolve the multimers, the genes that should be present inside the plasmid are the SUR sequences in case of Col1 plasmid. On the other hand, the gene or the proteins which are involved in breaking up these multimers, which are host coded, are coded by the XCR genes. So they code for certain proteins called ARG protein or PEP-A protein. What it does, that it binds to these SUR sequences and carries on the resolution of the multimers. The third mechanism of getting rid of curing of a plasmid or to prevent curing of a plasmid is the partitioning system. The partitioning system is a very important system that is present inside the plasmid, which makes sure that during cell division, each and every cell receives a copy of the plasmid. So this partitioning system makes sure that at least one copy of the plasmid should be retained within the daughter cell. This is supposed to be the most effective mechanism possessed by plasmids to avoid the loss of the plasmid during cell division. Next, the functions of these systems are regulated by a locus that is called as the PAR locus, where PAR stands for partitioning. PAR functions are also involved in the segregation of chromosomal DNA during cell division. So we may say that this PAR locus is also present within the chromosomal DNA as well. So herein, taking R1 as an example, we can see that the three important components are denoted as PAR C, PAR M and PAR R, where PAR C is a cis component or DNA component which is present within the plasmid itself as a DNA. On the other hand, PAR M and PAR R are protein coding genes. So herein we will see that PAR C part is present within the plasmid, wherein the first protein that comes and binds is called PAR R. And once PAR R binds to the PAR C DNA, then only PAR M, which is attached with an ATP molecule, it comes and it binds to the PAR R. And this complex between PAR C, PAR R and PAR M is responsible for segregation of the plasmid or partitioning of the plasmid during cell division. What we can see here, that PAR M can keep on polymerizing and they are getting attached, leading to release of a phosphate molecule. And ADP attached PAR M gets tied up and they polymerize, and this polymerization actually leads to separation of these plasmids and they push the plasmid towards the two poles of the cell. And finally, they break up and this PAR M moieties are further being recycled back and gets attached to another molecule of ATP to participate in second round of partitioning of a plasmid. So as because we have seen that this PAR mechanisms are operative, if these are operative and they are functioning properly, then the chances of the bacterial cell losing a plasmid during cell division is lost. So this is supposed to be the most strongest mechanism that makes sure that plasmids are not cured during cell division. Now we are going to discuss about another important topic regarding plasmids is incompatibility of a plasmid. As the term says that two plasmids are supposed to be incompatible, it means they cannot stay together inside the bacterial cell. So the concept of compatibility comes whenever we talk of existence of more than one plasmid inside one bacterial cell. Two plasmids are incompatible if and only if they share same replication machinery or they demand similar proteins for their replication process. As because both of them are residing inside the same host cell and they are demanding for the same types of proteins. So it becomes really difficult for the bacterial cell to understand the existence of two plasmids as two different entities. And what happens that as a result of which some of the plasmid gets replicated very nicely, and on that cost other one is depleted of replication, and during cell division we will see that gradually one of the plasmids is getting lost. Now the question arises that how you can determine the incompatibility of a plasmid. So first we will see that we are introducing a plasmid with a definite compatibility whose group is known to us inside a bacterial cell. Then we will try to introduce another plasmid into it, and we will see that whether the second plasmid is able to reside within the bacterial cell along with the first plasmid or not. If we see that they are able to reside together, then we will be saying that they are compatible. And if we see that one of them is getting lost, then we will be saying that they are incompatible, right? So next we will be discussing about incompatibility due to shared replication control.
Ma'am, what are the causes of incompatibility?
Generally, as I said, the main cause of incompatibility is that both the plasmids share same resources for their replication process. This is supposed to be the most important factor that regulates the incompatibility. And the second one is due to the copy number control mechanisms. As I said that that plasmids do have their own copy number control mechanisms, but if both the plasmids are alike, and if it happens that one of them reaches the desired copy number and the other fails to do so, then we will see that the one which is not able to acquire the copy number will be lost. So let's have a look that the two plasmids can be incompatible if they share the same mechanism of replication control. As I was saying, the replication control system does not recognize the two as different. This is the main thing, that it cannot differentiate them, and so either plasmid may be randomly selected for replication, and at the time of cell division the total copy number of the two plasmids will be the same, but one may be represented much less than the other. As because the both of them are sharing the same resources, although the desired copy number has been reached, but what will happen that one of them has reached a higher copy number at the cost of the other. This is the problem. So next here you can see that unequal distribution of the plasmids are occurring, but as because their requirements for replication are different, in the due course of time both of the plasmids are acquiring their desired copy number. On the other hand, here also there is unequal division of the plasmids, but herein what we can see that both of them are of the same replication requiring system. So what we will see that after completion of a certain stage in the life cycle of the bacteria, they will acquire a desired number, copy number. But the thing is, the total number is same in both the cells. But in one cell we will see that one plasmid is dominating, on the other cell we will see that the other plasmid is dominating. And gradually a time will be reached wherein we will see that some cells are containing the plasmid 1 and the other cell is containing the plasmid 2 only. And the next important thing or the causes of incompatibility is due to lack of partitioning system, right. So here we can see that the two plasmids can also be incompatible if they share the same partitioning system. So normally this helps ensure that both the daughter cells get at least one copy of the plasmid and neither daughter cell is cured of the plasmid. So this is the basic benefit of having a partitioning system. But if the coexisting plasmids are sharing the same par system, then one or the other is always distributed into the daughter cell. Means one cell receives one plasmid and the other cell is receiving the other plasmid. So the take home message is to avoid the loss of a plasmid because of incompatibility, one should be careful to select two plasmids requiring different replication mechanisms and different partitioning mechanisms. So in today's lecture, we have elaborately discussed regarding the definition of copy number, need to regulate the copy number and different factors that regulate the copy number. We have also discussed about the plasmid addiction system. We also elaborately discussed about the dimerization and multimerization, which are the causes of curing of plasmids and how plasmid actually prevents dimerization. We have also focused on the partitioning mechanism of the plasmid, and finally we highlighted on the incompatibility of a plasmid and causes of incompatibility.